Hello class, today we are going to learn about the microscopy and the microscopic techniques. This section is divided into two parts. There are principles of the compound light microscope and the instrument of microscopy. The most microorganisms are quite small. Protozoa is 100 microns. Bacteria is from 1 to 5 micron. Yeast is 8 micron. Rickettsia is 0.4 micron. Chlamydium and the microplasms are 0.25 micron, and viruses is 20 to 250 nanometer. Principles of compound light microscope, the Kohler and the objective lenses of compound microscope. With a compound light microscope, we can examine very small specimen as well as some of their fine detail, and it can contrast between bacteria and the surrounding. A series of finely grounded lenses form a clear, focused image that is many times larger than the specimen itself. This magnification is achieved when light rays from an illuminator, the light source, pass through the condenser, which has lenses that direct the light rays through the specimen. From here, light rays pass into the objective lenses, the lenses closest to the specimen. Most microscopes used in microbiology have several objective lenses. Measures light bending ability of a medium. Light may bend in air so much that it misses the small, high magnification lens. To achieve high magnification with good resolution, the objective lens must be small. Although we want light traveling through the specimen and the medium to refract differently, we do not want to lose light rays after they have passed through the stained specimen. To preserve the direction of the light rays at the highest magnification, immersion oil is placed between the glass slide and the oil immersion objective lens. Immersion oil is used to keep light from bending. Bright field microscopy are smallest of all optical microscopy illumination. Dark objectives are visible against the bright background. Dark field microscope used to enhance the contrast in unstained samples. The light objectives are visible against the dark ground in the dark field microscopy. The dark field microscope produces detailed images of living, unstained cells and organisms by simply changing the way in which they are illuminated. The dark field microscopy can reveal considerable internal structure in large eukaryotic microorganisms. Under usual operating conditions, the field of vision in a compound light microscope is brightly illuminated. By focusing the light, the condenser produces a bright field illumination. Microscopes are used to magnify small objectives. Most micrographs shown have size bars, symbols to help you identify the actual size of the specimen and the type of microscope used for that image. Because different microscopes have different resolution ranges, the size of the specimen determines which microscopes can be used to view the specimen efficiently. And this shows what Baker's yeast, flagella, and paramecium. The fluorescence microscope has become an essential tool in microbiology. Bacterial pathogens can be identified after staining with fluorochromes or specifically tagging them with fluorescently labeled antibodies using immunofluorescent procedures. In ecological studies, fluorescence microscopy is used to observe microorganisms stained with fluorochrome labeled probes and fluorochromes that bind specific cell constituent. In addition, microecologists use epifluorescence microscopy to visualize photosynthetic microbes. As their pigments naturally fluoresce when excited by light of specific wavelength, it is even possible to distinguish live bacteria from dead bacteria by the color they fluoresce after treatment with a specific mixture of stain. The principal use of fluorescence microscopy is the diagnostic technique called the fluorescent antibody technique, or immunofluorescent. Antibodies are naturally defense molecules that are produced by humans and many animals in reaction to the foreign substance or antigen. Fluorescent antibodies for a particular antigen are obtained as follows. An animal is injected with a specific antigen, such as a bacterium, and the animal then begins to produce antibodies against that antigen. After a sufficient time, the antibodies are removed from the serum of the animal. If the unknown bacterium is the same bacterium that was injected in the animal, the fluorescent antibodies binds to the antigen on the surface of the bacterium, causing it to fluoresce. Compares some of the important features of the light and transmission electron microscopes. The TEM has distinctive features that place harsh restrictions on the nature of samples that can be viewed and the meaning by which those samples must be prepared. It uses electrons, electromagnetic lenses, the fluorescent screens. Specimens may be stained with heavy metal salt. Specimens must be viewed in a vacuum because electrons are deflected by air molecules. Only extremely thin slices of the specimen can be viewed because electron beams are easily absorbed and scattered by solid matter. Among the most powerful microscopes are scanning probe microscopies. These microscopies measure surface feature of an objective by moving a sharp probe over the objective surface. The scanning tunneling microscope was invented in 1980. It can achieve magnification of 100 million times, and it allows scientists to view atoms on the surface of a solid. The scanning tunneling microscope have a needle-like probe with a point so sharp that often there is only one atom at its tip. The probe is lowered towards the specimen surface until its electron could just touches that of the surface atoms. Thank you for your attention.